I'd like to introduce Jessica again. She was an overachiever, as Amanda said, and did 2 projects this summer. So excited for you guys to hear about what she did with Soft Shell Clam. So come on up.
All right, I'll wrap this up and keep it a little bit shorter because we all know it's time for food soon after this. And all right, so this one's going to be a fun one. So pushing the limits, heat tolerance in soft shell clams. So first, what is a soft shell clam? It is the image on your left. So that is a soft shell clam, also known as a Maya arenaria. And the common names are soft shell clams and /or steamers. So it is found in the red area on the graph to your right from Maine to Virginia and has some of the highest concentrations in those points. So the economic value in Maine alone, it is a $5 to $10 million industry annually. And in 2019, the industry was valued at $54 million with wholesalers, distributors, and others. Okay, so the history in Maryland, they are native species to Maryland. So they were very popular in the 50s to 90s. As you can see, the blue line on this graph, that is the number of bushels that they actually caught during this time. So it was a very popular species to catch, but it started to decline due to overharvesting and other factors like that. And even though the value skyrocketed in the 90s, it is almost non-existent today. So some of the main species in the Chesapeake Bay currently that are commercially for the aquaculture industry are oysters, blue crabs, Atlantic menhaden, and other ones. So I'm going to focus specifically on oysters. So this graph to your right is the number of bushels of oysters in the Chesapeake Bay from the 1960s to 2006. And as you can see, the population size is decreasing. Like in Maya's presentation earlier, she talked about how global warming has been affecting the populations and causing a bigger spread of disease. So other factors like overharvesting, disease, and pollution are decreasing these populations. And so soft shell clams creates an opportunity that it creates a new species for aquaculture. So in case the oysters do bad this year, they have something else to fall back on. And furthermore, the soft shell clams are more tolerant and grow faster in low salinity conditions than oysters, which in Kayla's presentation, she mentioned all of the low salinity environments near here and how that can change very rapidly. And it reaches commercial size in 2 years or less. So some of the challenges for developing the soft shell clam culture in Maryland is seed production. So what happens normally for aquaculture industries is that they get the seed from hatcheries like the one we have in our actual Pearl laboratory. But soft shell clams, because this industry has not been big in a while, there are no seeds to do it. So they would have to go out and physically get their own clams by themselves, which would be hard to start out. And second, the culturing method, because usually in places like Maine, they have large stretches of land where you can actually put the soft shell clams in these intertidal zones. We do not have that opportunity, so we would have to start something from the ground up and actually develop something for our region. And third is the heat stress conditions. So they usually survive from -2 degrees Celsius to 28, but they prefer from 17 degrees Celsius to 23. However, in the Chesapeake Bay, they usually do not survive over 28. This creates a problem because just yesterday, it was over 29 degrees Celsius right in our pier. So it's definitely trying to find a species that can handle these waters. So our objectives are to achieve successful breeding, improve the heat tolerance, and selective breed based on phenotypic and genotypic breeding methods for heat tolerance. We've actually already achieved the first one. So Dr. Lou and Brittany Bryant were able to successfully spawn juvenile clams in our laboratory. And we now have around 468,000 juvenile clams. So we're just focusing on the other ones to get better. So the 2 approaches that we're thinking of using are phenotypic versus genotypic. Phenotypic would be we heat shock a large number of them and then take the survivors and breed that way. Genotypic is finding the specific genes that deal with heat tolerance to then use in better studies and to find the exact clams that have this. So my original project was going to be on the phenotypic approach, where we would heat shock around 600 adult and juveniles. However, this is the pier conditions and the temperatures of our pier right off of Pearl. And the blue line is when we actually took out the clams, and above the red line is above where they are comfortable. So that circle is the amount where the pier conditions spiked over this. So this has resulted in a huge mortality. So that left around 75 adults and 150 juveniles. So this was not an option at this time. However, we could still run the genetic approach to find those genes. So we still heat shock a smaller amount of these clams and sample the alive clams to get those genes. We then will send it to a lab to have their RNA sequence. And through these processes, we would identify the heat shock genes. We would identify the markers, and then this would help us specifically find the clams that we would like to breed later on. So what I did, we did a smaller heat shock thing. So the tank on the left is the control temperatures from 19 to 23 degrees Celsius. So these are the ones mimicking their ideal temperatures. And we have the tank on the right, which is heat stress, which was over 30 degrees Celsius, which would normally mean that this would kill almost all of them. So as you can see, there are also two different types of methods to actually hold them. So as you can see here, this is called a clam bottle, where we would place one clam in a bottle filled with sand. And then here are clam sandwiches, which I will show better here. So we also wanted to see which type of thing to hold them was better, because we don't have a specific method to develop these the best. So we're trying to find out which holding one would be better for it. So some pros for using the bottle would be that the sand could insulate and protect from challenging conditions by mimicking the natural environment. And it prevents from overcrowding because there's one clam per one bottle. So, and also it helps protect against accidents, because if you're going to be harvesting them, you need a large one. And if you accidentally drop them because you're dealing with hundreds of them, it would be better to have some insulation. For the sandwiches, the pros are they're so much easier to maintain and set up. You can have a higher density, there's a lot less prep work, and you don't have to deal with the sand. So some of the observations, the controls are on the left. It's a little hard to see in this light, but one of the things for heat stress, like right here, that is a siphon extended. So that's the filtering organ of these creatures. And here they're kind of more buried in the sand. So in the heat stress, more of the siphons were actually extended, but here you can see in the control, it's a little more open. So it's filtering better than the heat shock, where their siphons are a little more closed. Furthermore, their siphons responded slower to prodding when they were in heat stress. So they were very challenged by these conditions. Okay, so here is the percent mortality in the soft shell clams between the bottles and the sandwiches. We wanted to see which one was better. So in day 3, the sandwich actually had the first mortality, and it had a higher mortality rate consistently over all of our days. So they almost had 90+% that died in the sandwiches compared to the 60-ish% in the bottles. So our next step was, this was the genotypic approach I mentioned earlier. So we actually took the samples from the live clams. So the first death was measured on the 27th of July. And so we sampled from 3 clams from each group. So 3 from the sandwiches, 3 from the bottles from each tank. And so we took samples from 7 of the organs. So their gills, their heart, adductor muscle, foot, mantle, gonad, and siphon. We took those different samples to measure the different upregulation or downregulation of the heat shock genes in those different organs and see which ones we need to focus on for later research. And we also took another gill sample to actually have it be RNA sequence. So here is me and Dr. Liu going and actually taking the samples, and we processed the RNA in that lab that you saw Maya was in earlier. And so we got the RNA prepared for sending off to the lab. So in conclusion, in heat stress, the more clams in the sandwiches died. However, in the control, no clams died, so both methods work. So in the challenging conditions, the bottles might be a better alternative, but if you're going on a large scale and don't have challenging conditions, I would personally do the sandwiches because they were a lot easier to maintain and a lot more practical. So our future steps would be those samples you saw us take, send them to the lab, get the candidate genes back and validate them. And then we would use those genes to actually breed and pick some of the top clams that we have that have these genes. And then use this data to spawn the best individual to run trials on those offspring for later. We could also continue the bottle versus sandwich research to have a better method for other aquaculture industries. All right, and I would like to thank the Maryland Sea Grant and Morgan State University OTT IGAP grant. All right, here are my references. And are there any questions?
Go ahead with the Zoom comment / question. Can you make clam sandwiches on rye? Just kidding. If current climate change trends continue, how feasible would it be super clams that breed super clams who can withstand and thrive at even higher temperatures?
So if we can get as much, we will keep testing on the heat tolerance genes. If we are able to find the heat tolerance gene and actually manipulate it in the way we want, potentially we could go as high as we can for the changing conditions happening. Okay, I'll end it. Wait, can you go to the next slide, please? Also.
Yeah,
Okay.
should be able to.
So I'm going to graduate school soon. If anyone knows any programs or anything like that, I'm gonna major in biological sciences. I am interested in aquaculture and anything related to biology. So please, here is my email. You may take a picture and send me things.